Hello Road Watchers! Today we're going to be doing a couple of experiments where we're going to be extracting DNA.
Hi everyone, I'm Sydney, and today we are going to show how to extract DNA from a banana. So first of all, what is DNA? DNA is a double-stranded molecule that is basically the recipe book of our body. It contains everyone's unique genetic code and all our information.
DNA is what cells use to understand what proteins to express, where in the cell to express them, and at what times during the cell cycle those proteins should be expressed.
And so we are going to use a banana for today's experiment, but this would also work well with strawberries.
So I'm just going to spit into my clear, clean container until I've covered about the bottom layer of this container. I will see you back after I have produced all my spit.
So all you're going to need is 1/2 cup of warm water, 1 teaspoon of salt, 1/2 teaspoon of liquid soap, a strainer, a type of closable Ziploc baggie, and some cold rubbing alcohol.
For this experiment, you will need some dish soap, some contact lens cleaner, but you can also use pineapple juice or meat tenderizer, table salt, some toothpicks, a clean, clear container, and I have some rubbing alcohol that I've been chilling in my freezer. This is 91% isopropyl alcohol. You can find this at any store.
We'll go ahead and get started. First thing we're going to do is unpeel our banana and add it to the Ziploc bag. Try to make sure the air gets out of the bag. And then we are going to mash up our banana. Split it on open.
So I have put in enough spit to fill up the bottom of my container. I will save you from having to look at that too closely. But before we do anything with this, we first need to understand what I actually have here. What I have is just a collection of cells from my saliva. So while that means I do have DNA, I also have everything else that comes with cells that we're going to have to deal with before we can extract that DNA. So the first thing I'm going to have to address is the cell membrane. If you've studied the structure of the cell, you know that the cell membrane is a phospholipid bilayer that surrounds the cell and protects its internal environment from its external environment. To deal with the membrane, we are going to be using our dish soap to break through that membrane and break open, or lyse, the cells. So we're just going to take our dish soap, and we're just going to put in a couple of drops, just like that. And then I'm going to take my toothpick, and I'm just going to mix that around a little bit and make sure that that dish soap interacts with all of my saliva cells in that container and breaks or lyses open all of those cells.
Alright, once you get your banana nice and mashed in the bag, we're going to take our 1/2 cup of warm water and we're going to add 1 teaspoon of salt. After this, we're going to add 1/2 teaspoon of our liquid soap. And then we are going to stir our soapy, salty water solution. Alright, once our solution is nice and stirred, we're going to take our bag of mashed banana and we're going to pour it in there.
Now that we've broken those cells open, we have access to the DNA, but we also have access to the proteins that were in that cell before we open those up. So we need to break those proteins down. So when we precipitate out that DNA, we know that that's DNA and not DNA with some trace amounts of proteins in there. To do that, we're going to use our contact lens cleaner. And in contact lens cleaner, there are what are called proteases. Proteases are simply enzymes that can break down proteins, which is perfect here because it'll break down those proteins that were in the cell, but it won't break down the DNA. So we can be confident then that all we will be left with is some nice DNA. We won't get any of those proteins gunking up our sample. So now that I've added my detergent and I've added my protease, I need to add something to make sure that that DNA clumps up so when I precipitate it out, I can see it with my own eyes. To do that, we are just simply going to add some salt. If you've studied the structure of DNA, you know that DNA is a very negative molecule because it's made up of a sugar phosphate backbone, and those phosphate groups make that DNA negative. Salt, on the other hand, is made up of NaCl, and those Na ions are very positive. So by having those Na ions interact with the phosphate molecules, we can cause that DNA to clump up around itself and hopefully see that when we precipitate it out with the alcohol.
Just get that in there, make sure all the banana is combining with your solution. And then we're going to take our strainer and pour it in here. So now we're going to add our cold rubbing alcohol into the solution. And then we will wait a couple moments, and if you move the camera over the top, you should start to see some of the DNA. And that is how you extract DNA from a banana.
Awesome. So now that we've added our detergent, our protease, and our positive ions, now we get to do the fun part. So I'm going to put on my safety glasses for this because alcohol can be dangerous for your eyes. We're going to take out our chilled rubbing alcohol. Again, this is 91% isopropyl alcohol. And I'm just going to take this isopropyl alcohol and pour it slowly over my saliva mixture, making sure to not make too much of a mess with it. I'm just going to do this nice and slowly. You'll notice I'm tilting the container so the alcohol can kind of flow into the saliva, not kind of splash on top of it. All right. Then you can kind of just move your mixture around just a little bit to encourage those DNA molecules to float up into that alcohol. So what the alcohol is serving to do is it's serving to precipitate out the DNA. This works because DNA, while being soluble in the water base of our saliva, is not nearly as soluble in the alcohol. So that means when it comes up into that isopropyl alcohol that we just poured on, we can see all the clumps that we made with our extracted DNA. I have one right here. Let's take a look at it. All right, so we have our precipitated DNA right here. Let's take a look. So look at that right there. That is our precipitated DNA. And you can kind of see it's really nice and clumpy thanks to all those ions that we gave it. And we're just going, and I can even take my toothpick and kind of just bump it around just like that, manipulate it and move it all around just like that. Notice how it's very white, very wispy DNA. And how it tends to be sticking together right now. That has to do with the fact that we added in those ions and allowed it to clump up just like that. If you really want to, take it and take it out. Look at that. And that is exactly what your DNA looks like.